Hello, my students. Let's continue our lesson about traits and probability. As we said before, that Punnett square, we use it in order to determine the possible genotype of a certain cross. If we were examining the inheritance of only one specific trait, we call it a monohybrid cross. Let's take some examples, like if we used the cross between homozygous dominant crossed with homozygous recessive, heterozygous crossed with heterozygous, heterozygous crossed with homozygous recessive. The first example, the homozygous dominant crossed with homozygous recessive. So let's use Punnett square in order to determine what is the genotype of the offspring. So when we know that it's a homozygous dominant, so both of them will be written in capital letter. So this parent has homozygous dominant and the other parent is homozygous recessive. So both letters will be in small letter. Let's see. So the genotype of the offspring might be F capital F small in this box, or F capital F small, or F capital F small, F capital F small. So this F during meiosis, each allele will be separated. So it will donate, this parent will donate F capital to each one of their offspring and 100% of their offspring, and the other parent, because it is homozygous recessive, so it will donate one F small to all of their offspring. So 100% of the offspring will be heterozygous, so they will have the purple color. So the genotype is 100% heterozygous and the phenotype 100% purple. So all offspring receive a dominant allele from one parent and recessive from the other parent, so 100% heterozygous and 100% purple in color. But if we crossed a heterozygous with another heterozygous, like in this picture, heterozygous purple with heterozygous purple, here we can see difference in the genotype of the offspring. So from each parent, half of the offspring receive a dominant allele, while the other half will receive a recessive allele, which will result in 1/4 of the offspring will have a homozygous genotype for the dominant trait, and 1/2 will have heterozygous, and 1/4 will have the homozygous recessive. So 1/4 dominant homozygous, 1/4 recessive homozygous, and half will be heterozygous. So in other words, the genotype ratio is 1 to 2 to 1, homodominant to 2 heterozygous to 1 homozygous recessive, and the genotype ratio is 3 to 1, 3 purple to 1 white flower. The third example is if we crossed heterozygous with homozygous recessive. So there is a chance, as we can see, that all offspring will receive a recessive allele from the homozygous recessive parent, and half of the offspring will receive a dominant allele from the heterozygous parent, and the other half will receive the recessive allele. So we will end up having half of them will be purple in color and half of them will be white in color. So the phenotype ratio will be 1 to 1, one purple to one white, and also the genotype will be one heterozygous to one homozygous recessive. But supposedly, we don't know if we had a purple flower and we don't know its genotype. So we picked up a purple flower and we don't know if it's pure homozygous purple or heterozygous purple. So what we can predict its color if we crossed it with a white flower. So if we crossed it with a white flower, chances are if it was a homozygous dominant, then all of the offspring will be purple in color. But if the offspring, half of them were purple while the other half is white, then this means that this purple flower was heterozygous. So crossing a homozygous recessive organism with an organism of unknown genotype, we call it test cross. So we use test cross in order to determine the genotype of an unknown organism. Thank you.